Welcome back to the deep dive. We're here again, digging into the sources to find those real aha moments, the ones that kind of shift how you view things. And today, we are diving deep into something just fundamental. It's everywhere in life, totally vital, but maybe we don't think about it much. Enzymes. Yeah, enzymes. You can think of them like, I don't know, the unsung heroes, the master architects running all the chemical reactions inside you, inside every living thing. So, our mission today isn't just what they do, it's how. How do these amazing little proteins work within those super strict rules of thermodynamics? How do they speed everything up? And how do they manage to make things happen that seem like they shouldn't? Get ready, because we're going to explore how these tiny powerhouses basically run the show, making reactions happen precisely when needed, like right now, inside your body. Okay, let's unpack this then. Enzymes. You immediately think energy, chemical reactions. But like you said, the universe has these strict laws. Thermodynamics, how do enzymes fit in? Do they get a special pass? Or bend the rules somehow?
That's exactly the right place to start. Because the really cool thing is enzymes absolutely obey those laws. They don't cheat physics at all. Think of it like trying to run a factory where every single step has to go downhill, energetically speaking. That's thermodynamics for you. Enzymes are masters at working within that framework.
Oh, okay, working within it. So they're not breaking laws.
Not at all. What they can do, specifically, is increase the speed of reactions that are already energetically favorable. We call these spontaneous reactions. Now, spontaneous doesn't always mean fast. It just means given enough time, maybe even millions of years, they would happen on their own because they release free energy. It's like a ball rolling downhill. It naturally wants to do that, releases energy, and actually increases the disorder, the entropy of the universe. That release of energy is measured as a negative delta G.
Right, negative G, I remember that. So that's the downhill reaction, releasing energy, heading towards more disorder. And the flip side is positive, that's pushing the ball uphill, needing energy put in. And you're saying enzymes on their own just can't make that uphill push happen.
Exactly. And it's a really fundamental constraint. Enzymes cannot force an energetically unfavorable reaction, one that needs energy input, a non-spontaneous one, to just happen by itself. They aren't magic wands.
Yeah.
They can't conjure energy from nowhere or, violate the second law of thermodynamics. If a reaction is uphill, thermodynamically speaking, an enzyme alone isn't going to cut it.
Okay.
So it's vital to get this. Enzymes make the possible happen faster. They don't make the impossible possible. Well, not directly anyway.
That's such a key point for you listening. Enzymes aren't breaking the rules, they're exploiting them maybe. They accelerate what can happen. Okay, that clarifies things a lot. So if they're not forcing impossible reactions, how do they speed up the favorable ones so incredibly much? It's not just like shaking the test tube harder, not just random heat.
Oh, definitely not. It's way more elegant, much more specific than just bumping things together randomly. The secret sauce really is that enzymes lower the activation energy of a reaction. Think of it like this. Even a reaction that's downhill overall, one that releases energy, it still usually has this little energy hump it needs to get over just to get started.
Ah, okay. Like needing a little nudge to get going, even if the path slopes down afterwards.
Precisely. Even rolling a ball downhill often requires that initial push over a little ridge first. That ridge is the activation energy barrier. Without help getting over that hump, even a favorable reaction might just sit there for, ages.
So the enzyme gives it that nudge, but how?
Through some really clever molecular choreography, you can picture them as tiny, highly specific machines or maybe matchmakers. First, an enzyme grabs its specific reactive molecules, we call them substrates. It binds them in a special pocket, the active site, holding them in just the perfect position relative to each other, like ensuring they shake hands exactly right. This alone massively increases the odds of them reacting.
Okay, so it lines them up.
Lines them up perfectly. And second, during the reaction, you often get these really unstable temporary structures, chemicals called transition states or intermediates, or like halfway points. Enzymes are brilliant at stabilizing these fleeting high-energy moments. Imagine holding a wobbly bit of a model steady while you attach the next piece. That's kind of what the enzyme does. It lowers the energy needed to reach that transition state, guiding the reaction smoothly towards the final products.
Okay.
And the result, the reaction happens on demand, controlled, step by step, and often like a trillion times faster.
A trillion. Wow. That's almost impossible to imagine. Okay, so this is where it gets really interesting for me. It's that precision, that control. It's not just brute force heat. It sounds so much smarter for, a living cell. What's the big advantage of this controlled approach, biologically speaking?
Oh, the advantages are huge, foundational, really. For one thing, because it's so precise, you lose much less energy as wasted heat. Think about it, if every reaction in your body just dumped heat uncontrollably, you'd basically cook yourself. Enzymes let complex chemistry happen efficiently at body temperature. That's amazing engineering.
Right. Avoids overheating. Makes sense.
And this efficiency leads to something else really crucial. Instead of all the released energy just dissipating, a good chunk of it, maybe around 5% from these favorable reactions, is actually captured, stored.
Only 5%. Doesn't sound like much.
But in biology, 5% captured consistently is enormous. That captured energy often gets funneled into making molecules like ATP, adenosine triphosphate. That's like the cell's rechargeable battery, its main energy currency. And that stored energy in ATP. It's then used later to power other things, like building new molecules by forming different covalent bonds. Those are the strong links holding molecules together. So it's constantly recycling energy to build stuff.
Okay, wow. So that 5% is like the cell's operating budget, constantly being topped up and spent. That's clever. And just to double check, the enzyme lowers the activation hump, speeds things up, but the overall energy drop, the adhere from start to finish, that stays the same.
Spot on. Absolutely the same. The starting point and the ending point energetically are unchanged. The enzyme just provides a super fast, efficient highway between them instead of a slow, bumpy track.
Got it. Okay, so that leads to the big question, doesn't it? If enzymes can't force those uphill reactions, the ones that need energy, how does life actually do anything complex? Building proteins, growing, moving muscles? All that requires energy input. It seems like it needs to go uphill. So how? There has to be a trick, right? Because cells obviously manage it.
You've nailed it. This is where biology pulls off one of its most brilliant tricks, something called reaction coupling. And yes, enzymes are right at the heart of this, too. They often play a kind of double role here.
A reaction coupling. Okay, what does that involve?
Well, in these cases, the enzyme is often facilitating two things at once. First, it still does its usual job, lowering the activation energy for all the reaction steps involved, both the uphill and the downhill parts. But second, and this is the really ingenious bit, it physically links the energetically unfavorable reaction, the uphill one, to a separate, highly energetically favorable reaction, a downhill one that releases a lot of energy.
Links them, like ties them together somehow.
Exactly like that, at a molecular level. Imagine you need to lift a heavy bucket up. That's your unfavorable reaction, needs energy. But next to it, a much heavier weight is falling down. That's your favorable reaction, releasing energy. If the enzyme acts like a rope and pulley system, linking the falling weight to the bucket you want to lift, the energy released by the weight falling can be used to pull the bucket up.
Ah, I see. One reaction pays the energy cost for the other one.
Precisely. We look at the overall energy change, the summative D. You take the positive G of the uphill reaction and add the negative GG of the downhill reaction it's coupled to. If the total sum is negative, meaning overall, the combined process still releases energy and moves towards disorder, then the whole coupled reaction can proceed spontaneously thanks to the enzyme linking them. The energy release from the favorable one drives the unfavorable one.
So the downhill reaction has to release more energy than the uphill one needs.
Exactly. The net change must be negative. A classic example is breaking down glucose sugar. That releases a lot of energy, a big negative ATT. Enzymes couple that release to the creation of ATP, which requires energy, a positive AD. The breakdown of glucose pays for the making of ATP. Then that ATP carries the energy off to power muscle contraction or build proteins or whatever else needs doing. It's this constant enzyme-managed energy transfer.
That's incredible. It's not just a trick. It's fundamental to how life builds complexity. It's like an energy economy inside the cell managed by enzymes. And the enzyme itself, after orchestrating all this, it just steps back, unchanged, ready for the next reaction pair.
Absolutely. That's another beautiful part of it. The enzyme is a true catalyst. It facilitates the whole process, lowers the barriers, couples the reactions if needed. But it emerges at the end exactly as it started. It's not used up. It's ready to go again, potentially millions of times a second. This reusability is essential. Imagine if cells had to make new enzymes for every single reaction. The energy cost would be astronomical.
So, okay, wrapping our head around this, what does it all mean for you listening? We've seen enzymes aren't just making things fast. They're these incredibly sophisticated molecular machines. They work strictly within the laws of thermodynamics. They lower those activation energy hurdles masterfully. And maybe most cleverly, they enable all the complex building blocks of life through this ingenious reaction coupling. They really are the engineers making life work, second by second.
And understanding this, even just the basics, gives you a real insight into yourself. It explains how your body can perform millions of different precise chemical reactions constantly. Thinking, moving, healing, just being. It all relies on this incredibly efficient energy management system orchestrated by enzymes. It's the elegant principle underlying almost everything your body does. Without them, life would be impossibly slow, chaotic, just unworkable.
So maybe next time you feel that burst of energy running for a bus or even just digest your lunch or, marvel at a plant growing, take a second. Think about the silent, tireless work of these molecular matchmakers inside and maybe wonder what other incredibly elegant energy coupling processes are happening all around us or inside us, working perfectly, just waiting for us to notice. There's still so much amazing chemistry to explore. Thanks for joining us on this deep dive.